Joyce at San Gabriel, and for today, I am going to present our experiment about digestion of proteins. The experiment 21. Pepsin breaks down proteins into smaller peptides. It is produced in the chief cells of the stomach lining and is one of the main digestive enzymes in the digestive system of humans and many other animals, where it helps digest the protein in food. So as we can see in the first table, it shows the result of the gastric digestion of proteins using pepsin through Biret test. The first test tube contains 1 pinch of egg white, 3 ml of pepsin solution, and 3 ml of water. The second test tube contains 1 pinch of egg white, 3 ml of pepsin solution, and 3 ml of 0.1 mole of hydrochloric acid. Lastly, the third test tube contains 1 pinch of egg white and 3 ml of water. In neutralizing 1 ml of each hydrolysate with solid sodium carbonate, the first and the third test tube yields color violet solution, while the second test tube yields a color pink solution. On the other hand, the second table shows pancreatic digestion of proteins using pancreatine through Biret test also. The first test tube contains 1 pinch of egg white, 3 ml of neutral pancreatine, and 3 ml of water. The second test tube contains 1 pinch of egg white, 3 ml of neutral pancreatine, and 3 ml of 0.5 mole of sodium carbonate. And the third test tube contains 1 pinch of egg white and 3 ml of 0.05 mole of sodium carbonate. In neutralizing 1 ml of each hydrolysate with solid sodium carbonate, the first and the third test tube yields color violet solution also, while the second test tube yields color blue solution. So now, the significance of the Biret test. It is a chemical test used for detecting the presence of the peptide bonds. The Biret test is important mainly because it can detect the amount of protein in urine. The violet color is a positive test for the presence of protein, and the intensity of the color is proportional to the number of the peptide bonds in the solution. Next, in digestion of proteins using pepsin, the test that can indicate the completeness of protein digestion is Biret test. Pepsin will only give a pink color in Biret test because it can only break the carboxyl group of aromatic rings of leucine and alanine. While in digestion of the proteins using pancreatic protease, the test can be Milon's test, nitroprusside test, Hopkins-Coe test, Santoprotic test, Sakaguchi test, and Poli test. Pancreatic protease will get blue color because of higher chance, since it can extract more enzyme such as trypsin, chymotrypsin, and carbopeptidase. Okay, the factors needed in the gastric digestion of proteins are concentration of the enzyme, which is the protease that breaks down proteins into smaller chains, the amount of protein food needing action, the acidity of the food and of the stomach, the temperature of the food, time, and presence of any digestion inhibitors such as antacids. The role of hydrochloric acid in the stomach denatures salivary amylase and other proteins by breaking down the structure and thus the function of it. The hydrochloric acid also converts pepsinogen to the active enzyme pepsin. Pepsin is a protease, meaning that it cleaves bonds in proteins. Sodium bicarbonate is secreted in the pancreas to help aid in digestion. Its role is to help neutralize stomach acid generated during the digestive process and breaks down the certain enzymes. Pancreas contains exocrine glands that produce enzymes important to digestion. The factors needed are acidity and alkalinity of the environment, presence of inhibitor compound, and temperature. Next, the enzyme involved in protein digestion. First, the pepsin. Pepsin is a protein that produced in the stomach and is one of the main digestive enzymes in the human digestive system. It works in conjunction with chymotrypsin and trypsin released in the small intestine. It is also a digestive enzyme that chemically digests or breaks down proteins into shorter chains of amino acid. Second is chymotrypsin. It is a digestive enzyme that breaks down proteins and also be referred to as protease. It is naturally produced by the pancreas in the human body. Third is the trypsin. It may also be referred to as proteinase. It is an enzyme that helps us digest protein. In the small intestine, trypsin breaks down proteins, continuing the process of digestion that began in the stomach. Trypsin is produced by the pancreas in an inactive form called trypsinogen. Thank you so much guys for listening.